Hello students, this is Kanika. Welcome to my channel PrepBiology. So today in this lecture we are going to discuss top 20 previous year questions from your chapter number 1 of biology, that is the sexual reproduction in flowering plants. Okay, so these all are the previous year's questions which have asked repeatedly in your previous class 12th board exams. Okay, by this lecture we will also discuss all the important topics of your chapter, that is sexual reproduction in flowering plants. Let's begin with the lecture. So the first question is, which have asked in your 2024, 2020 and 2019 years board exams, draw the TS of anther. This TS of anther structure is very important, and label its wall layers, different wall layers, and mention their function. This is the most important topic from your chapter number 1. That is the TS of anther. As you all can see, this is the TS of anther and having different layers: epidermis, endothelium, middle layers, and having the MMCs and the tapetum. Okay, so these all are the wall layers, and this is the structure of the TS of transverse section of anther. This structure is very important. Okay, so if we see the chart, this chart we have discussed in the chapter number 1 classes. Okay, so we have the different layers of the anthers and having the different characteristics of each layer. Composition is a different layer, and the function of each layer is also very important. Like epidermis, it helps in protection. Endothelium, it prevents splitting and bursting of the pollen. Middle layers helps in the pollen production, and tapetum, which is the most important layer of the anther that nourishes the developing pollen grains, provide the nourishment. Okay, so this chart is itself is very important. The MCQs and the PYQs have asked in your previous years. Next question is, where the following structures present in the male gametophyte of an angiosperm? You have the different structures like germ pore, sporopollenin, and generative cells. Okay, so you all have to mention that these structure, what is the importance of these structure and where they are present. So germ pore, we know that in the pollen structure we have the outermost layer is the axine, inner is the intine, inside it we have the nucleus, and in the axine part, in the axine, that is the outer layer of the pollen, having the germ pore, okay, which is present in the axine of the pollen grains, and it is the place where the axine and the sporopollenin is absent. Okay, the germ pore where the sporopollenin is absent, that we have discussed in the classes also. Okay, so next is the sporopollenin. What is sporopollenin? Sporopollenin is present on the outer surface of the pollen grain. Okay, the axine is made up of the sporopollenin, okay, and it is the one of the most resistant organic material known. Okay, and it can withstand high temperature and strong acids and alkalis. No enzyme is found that degrade the sporopollenin. In the many adverse conditions of the plants, the axine helps the pollen to withstand in higher temperature or the strong acids or alkalis. Why? Because this sporopollenin is itself is the most resistant component. Okay, and no enzyme will degrade this. Okay, next is the generative cell. Where the generative cell is present, we know that when the pollen having the multiple divisions occurs, we have at last two celled stage, that is the vegetative cell and the generative cell. Okay, and then generative cell will form the two male gametes. Okay, so this is present in the pollens. These type of questions I have also asked in your previous years. Next question is, explain the process of development of the male gametophyte. This is a very most repeatedly asked question. Okay, why is it called male gametophyte? Okay, so the development of male gametophyte, these are the diagrams we have discussed in the classes. That first, the sporogenous cells present in the anthers, it will mature and form the MMC, that is microspore mother cell. Microspore mother cell, one microspore mother cell is diploid in nature and it will undergo meiosis 1. They will form the dyad, and meiosis 2 occurs. They will form the microspore tetrad. 4 microspores are present. So, one of the microspore, that is the pollen grain having the axine intine, they will mature into the, and asymmetrical division occurs and forms the vegetative cell and generative cell. So these two diagrams are very, very important for the male development of the male gametophyte. Why is it called male gametophyte? Because at the end this generative cell will form the two male gametes, that's why it is called the male gametophyte. So the development of the male gametophyte, the structure, the process is also very, very important. Next question is, where does microsporogenesis occurs in an organism? So we know that microsporogenesis, this is the microsporogenesis, okay, where the microspores are formed. These are formed in the anther part, okay, anther of a flower, okay. How is a three-celled male gametophyte different from it? So we know that there are the two kinds of the male gametophyte, that is the two-celled male gametophyte and three-celled male gametophyte, okay. This is the two-celled male gametophyte. Why? Because the pollen grains, when the asymmetrical division occurs in the pollen grains, the vegetative cell and the generative cell, two-celled stage is formed. Okay, two-celled, one is vegetative and second is generative. What is three-celled stage? When this pollen attached on the surface of the stigma, this is the pollen, it is attached on the surface of the stigma. This is the stigma part of the ovule. This is the pollen, okay. This is the pollen, and it will emerges the pollen tube, okay. And when the pollen tube forms, this generative cell will divide into two male gametes, okay. So we have one is vegetative cell. This is the vegetative cell, and generative cell is divided into two male gametes, and these are the two male gametes. How many cells are there? 3 cells are there. So this is the three-celled stage, and this is the two-celled stage. The difference between the three-celled and the two-celled stage of the male gametophyte is very, very important. Okay. Next question is, draw LS of pistil. LS of, that is the longitudinal section of the pistil showing pollen tube entering into the embryo sac. Label the following. You have the mentioned the different structures like micropyle, synergid and nucellus. You all have to draw the LS of pistils. Okay. So this is the LS of pistil or the ovule having the nucellus. Nucellus is present and antipodal cells and synergy cells, and we have the two ends. We know that we have discussed the structure of the ovule or the LS of the pistil. We have this kind of structure is there, okay. This is the LS of pistil, okay, or the ovule. So we have, you know that this is the pollen sac. Oh, sorry. This is the embryo sac. Okay. Outside the embryo sac, you have the mass of perigametous cells. That structure is called the nucellus. Okay. What are antipodals? Antipodals, when the embryo sac is developed or mature, the antipodals are formed. There is the 3 antipodal cells, and at the, this is the micropylar end, and this is the chaliceal end. Okay, and at the micropylar end we have the 2 synergids and one is axil. This is axil and this is the 2 synergid cells. Okay, and the position of the micropylar end and chaliceal end is clear to you all. At the chaliceal end, the antipodal cells are present. At the micropylar end, the synergids and the egg cell is present. That is called the egg apparatus. Okay, and in the middle of it, we have the polar nuclei. So you all have to know that these different structures, where these different structures are present, and the function of each and every structure is also very, very important. Okay, so the egg cell is important for double fertilization. Nucellus give the nutrition for the developing embryo sac. Synergids also helps in nutrition, or it helps in the entry of the pollen tube inside the egg apparatus. Okay, and micropylar, and it also give path to the pollen tube for the entry of the to the embryo. Okay, so these all functions and this structure of the LS of ovule is very important. Next question is, explain, this is the monosporic development of the embryo sac in an ovule of an organism. So this question have asked in your 2023 board exams and 2021 board exams. Okay, so monosporic development, when the one spore is converted into the development of the embryo sac. Mono means single. So we know that this structure is very, very important, shows the monosporic development. Why? We know that the microspore dyad, when it converted into the four, that is megaspore tetrad, megaspore dyad is converted to megaspore tetrad, 4 cells are formed, and out of 4 cells only one is functional in the megaspore, and it will divide further and form the embryo sac. Okay, so this is the structure that comes under the monosporic development of the embryo sac in an ovule. Okay, and draw the diagram of mature embryo sac, and this is the diagram of mature embryo sac, where we have the chalazal end, micropylar end. Chalazal end have antipodals, micropylar have the egg apparatus, we have the polar nuclei at the middle. So these all structures are also very, very important. Revise these structures carefully. Okay. Next is a mature embryo sac in a flowering plant having 7-celled 8 nuclei stage. Explain with the help of the diagram only. So we know that one that is the 7-celled 8 nucleate stage. So by the help of diagram, you all have to answer this question. So 7-celled 8 nucleate stage, we know that 7-celled, how 7 cells are formed? 3 cells are antipodals. Okay. Next is 1 cell is egg cell, 2 cells are synergids, 3 plus 3 is 6, and 1 is the central cell. So 7-celled is 3 is antipodal, 1 is central cell, 1 is egg cell, and 2 is synergids. 3, 4, 5, 6, 7. So this is the 7-celled. 8 nucleate. So 8 nucleus are there. 3 antipodal nucleus. Okay. 3 egg apparatus nucleus. 1 is egg cell and 2 are of synergid. So this is 3. And 2 polar nuclei are there. So we have 3 plus 3 plus 2. 6, 7, 8 nucleate stage. So this is the diagram in which we can see the 7-celled 8 nucleate stage. Okay. So this diagram is also very important. Next question is, name any 2 outbreeding devices. This is the recent year question of previous year, that is 2024. This question have asked that name any 2 outbreeding devices that flowering plants have developed and explain how they help in encouraging the cross pollination. So outbreeding devices you all have to name and explain how they encourage the cross pollination. Okay. So we have studied there are different outbreeding devices like declining, self-sterility, dichogamy. Okay, so these all are the outbreeding devices. What are these devices are? Declining means unisexuality, means that the only single sex organ is present, whether it is a female or male. Okay, or we can say that males and female sex organs are present in different flowers. That is the unisexuality. The flowers are unisexual, so the self-pollination is not possible in this condition. Okay, examples are mulberry and papaya. Okay, so if the self-pollination is not there, then of course it will encourage the cross-pollination. Okay, second device is the outbreeding device is the that is the self-sterility. Okay, self-sterility or the self-incompatibility means the pollen of a flower do not germinate on the stigma of the same flower due to the presence of similar self-sterile genes. So, if the male and females are present in the same flower, but the pollen, that is the male flower, do not germinate on the stigma of the same flower due to the presence of the similar sterile genes, that is called the self-sterility, or sterile means fertility. Okay, so that is called the self-incompatibility. Examples are tobacco, tomato, crucifers, etc. So the examples of each and every term is very important. So make sure you will note it down carefully. Next is dichogamy. Dichogamy means anthers and stigma mature at different times in bisexual flower. So in the bisexual flower, that is when the in the single flower there are male and female both are present, but they mature at different times. That is the dichogamy, and it will of course encourage the cross pollination. Okay, so this is important. The examples are also very important. Okay, so these are the some of the outbreeding devices. Outbreeding devices are developed to avoid the self-pollination and encourage the cross-pollination. These are the outbreeding devices. What are outbreeding devices? Which avoid the self-pollination and encourage the cross-pollination. Okay, so these are also very important. Next question is, explain the mechanism of pollination in marine sea grasses like Zostera, or in many years you have the question like express the process of pollination in Vallisneria. The Vallisneria, Zostera, these all are the examples which are also mentioned in your NCERT biology textbook. So make sure that you all understand it carefully. Okay, so these examples are also very important from your pollination topic. So we know that Zostera and Vallisneria, this is the plant of the Zostera and this is the Vallisneria. Okay, so we know that these are the aquatic plants, okay, which are live in the water. So the which type of pollination is there? Hydrophily. Okay, hydrophily means the water pollinated flowers are there. So in the Zostera, this is the Zostera, hydrophily occurs and the female flower remains submerged. Okay, so the female flower remains submerged in water and pollen grains released inside the water. Okay, and the pollen grains in such species are long, ribbon like, and they are carried passively inside the water. Some of them reach the stigma and achieve the pollination. Okay, so if we see the Vallisneria and the Zostera, the process of the pollination is same. How? Because in both the cases this is the female plant and which is submerged in water, but in the male plant the pollens are released in water and it stick to the stigma of the female flower. Okay, so these both are the plants in which the water pollination occurs, and in the Vallisneria plant, the these pollen grains are covered by the mucilage covering, okay, which helps in sticking the stigma as well as protect from the wetting by water. So these are some of the features of the pollination of both the plants Zostera and the Vallisneria. The structures are also very important. Next question is from your one of the major approaches of the crop improvement program is artificial hybridization. Okay, Artificial hybridization. Explain the steps involved in making sure that only desired pollen grains pollinate the stigma of a bisexual flower by the plant breeder. So, what is artificial hybridization? It is one of the major approaches of the crop improvement program. Why? In such crossing experiments, it is important to make sure that only the desired pollens, okay, in this the desired pollens grains are used for the pollination and stigma is protected from the contamination. Okay, so this is achieved by in artificial hybridization. We artificially hybridize the male and the female plant, okay, by the two main approaches. Like first is the emasculation, second is the bagging technique. What is emasculation? Emasculation is the removal of anthers. Okay, so we remove the anthers from the female plant, okay, and we artificially hybridized with the other male plant. By this, the pollen grains pollinate the stigma of the bisexual flower by a plant breeder. Okay. Second technique is the bagging. Okay. The bagging involves the covering. This is the covering by the polythene bag or the butter paper in order to protect them from the contamination by the foreign or undesirable pollen. So these are the two techniques which are comes under the artificial hybridization and they helps in the desired pollen grains pollinate the stigma of the bisexual flower by a plant breeder. So these emasculation and bagging techniques are important. Okay. Next question is the differentiate between geitonogamy and xenogamy. Okay. So we know that we have discussed the different types of the pollination in the pollination topic. If you want to check out that class, I will mention the class link in your description box. You can check out from that link. Okay. So pollination types, we know that self-pollination and cross-pollination. Under the self-pollination, we have the autogamy and geitonogamy. Autogamy is further divided into homogamy and clistogamy. Okay. And cross-pollination, which is also called the xenogamy, having the different biotic and abiotic agencies, in which we have discussed the different hydrophily, anemophily, entomophily, chiropterophily, ornithophily, etc. So this chart is very, very important, and difference between each and every subpart is also important. So in your 2019, 2020, 2021, so these questions have asked that the differentiate between geitonogamy and xenogamy. What is geitonogamy? It is the pollination between two flowers of the same plant. Okay. Xenogamy is cross-pollination. It is the pollination between two flowers of different plants. The flowers are genetically similar. Xenogamy genetically different. So geitonogamy comes under the self-pollination and xenogamy comes under the cross-pollination. What is clistogamy? Write the advantage and disadvantage. Clistogamy is where we have studied this. This comes under the autogamy. Okay. So clistogamy is the condition where pollination occurs in closed flower that do not open at all. And these are the advantages. Advantages is the seed setting is assured even in the absence of pollinators, as pollen on maturity will always reach the stigma due to their close placement. And disadvantage is it does not allow cross pollination, thereby restricting chances of the genetic variability. So these kind of questions have asked the pollination topic, and these are the types of the pollination is very, very important. The next question is explain any 4 devices that flowering plants have developed to encourage the cross pollination. Okay, so we know that which devices encourage the cross pollination. These are devices under the outbreeding devices. Okay, we have discussed already, like declining, self sterility, dichogamy. Under the dichogamy we have the 2 conditions, protogyny and protoandry. Okay, protogyny and the protoandry. Okay, so that we have discussed. Okay, so the next question is, why do plants discourage the self pollination? State any one reason. Because we know that in the self pollination, same flower or the genetically same gene, the flowers with the same genes or the genetical makeup, where the genetic makeup is the same, that that type of the pollination occurs, that is the self pollination. Okay, so why it will discourage? Because continued self pollination will result in the inbreeding depression. Inbreeding depression means in a small population, mating between the relatives are common. Okay, and the inbreeding may lower the population. Inbreeding may lower the population's ability to survive and reproduce. Okay, survive and reproduce. Okay, so no variations or variabilities have shown in the generations. So the self-pollination will result in inbreeding depression that will lead to the lower the population's ability to survive and reproduce. That's why the plants discourage the self-pollination. Okay. Next is explain the process of double fertilization. This process is very important. Have multiple times asked in your previous years. Okay, double fertilization. Why does the development of endosperm precedes that of the embryo? So the first is double fertilization. We know that there is a double fertilization in which first fertilization, male gamete fused with egg cell form the zygote. These are the end product, and the ploidy of the zygote is 2n, that is diploid. Second fertilization, triple fusion occurs, male gamete fused with the two polar nuclei that will form the endosperm. The ploidy of the endosperm is the triploid, that is the 3n. Okay, so these structures are also very important. Sometimes you have mentioned these structures and labeling, you have to label x, y and z. Okay, so in this structure the male gamete enters through the filiform apparatus, and in this structure the male gametes totally enters into the egg apparatus and fuse with the egg cell and the polar nuclei. That will show the double fertilization. Okay, why does development of endosperm precedes that of embryo? So the answer of this question is that when the endosperm is formed from the double fertilization process, okay, so the cells of these tissues, the endosperm cells are filled with the reserved food material, okay, and which is used for the nutrition for the developing embryo. Okay, so that gives the nutrition to the developing embryo. That's why the development of the endosperm precedes that of embryo. Okay, so this is the answer. So this is the primary endosperm cell, PEC and PEN. This structure is also very important. In your 2022, I think this structure have given for the question, and you have to label the different parts. Okay, so PEN, that is primary endosperm cell, and primary endosperm nucleus, they will form the zygote and having a different embryonic development. That zygote having a converting to the globular embryo, then heart-shaped embryo, then we have the complete dicot embryo. Okay, so these all structures are very important. Next question is the differences of this unit is also very important, which have also mentioned in your NCERT exercise, that difference between perisperm and pericarp, difference between syncarpus pistil, apocarpus pistil, difference between hypocotyle, epicotyle. Okay, so these are all difference. Just go through these all differences I have mentioned in the classes also. Perisperm, this is the perisperm is the layer that is a unused nucellus in the seed. This is the seed covering, and pericarp is the fruit covering. The difference is important. Apocarpus, syncarpus ovary. Apocarpus, the flowers has several free ovaries, and syncarpus is fused when in the fused condition. Okay, the difference is also important. Hypocotyle, epicotyle. Hypocotyle, the portion of the embryonal axis which lies below. Hypo means below the cotyledon in the dicot embryo. Epi is upper or above which is present above the cotyledon in the dicot embryo. Okay, so the difference of each and every part, that is perisperm, pericarp, syncarpus, apocarpus, true fruit, false fruit. Okay, so these all differences are repeatedly asked in your previous classes. Next is the question, that this question is recently asked in your 2024 board exams. So the, why do the plant seeds like viola and oxalis, these examples have mentioned in your NCERT textbook. So the NCERT itself, this textbook is very important for your board exams. Viola and oxalis give assured that seed sets even in the absence of pollination. So these are the flowers of viola and oxalis. Okay, viola and oxalis. So these are the examples of clistogamous flower. Okay, in one of my YouTube lecture, I have mentioned the difference between clistogamous flower and chasmogamous flower. Okay, so clistogamous flowers are the flowers which do not open at all, and anthers and stigma lie close to each other. Okay, so this is the flower of the viola and this is the flower of the oxalis. Okay, so the viola and oxalis give assured they form the seed sets without the presence of the pollinators. This is the question, ki why they showed the seed set even in the absence of pollinators, because the viola and the oxalis are the clistogamous flower. So through the process of clistogamy, in which the flowers do not open at all and anthers and stigma lie close to each other. Okay, and when the anther dies in the flower buds, pollen grains come in contact with the stigma to carry out the pollination. So this is very important. The clistogamy ensures no chance of cross pollen. Okay, it ensures that no chance of cross pollen landing on the stigma, okay, and produce ensured seed sets in the absence of the pollinator. So viola and oxalis, if you want to check out the difference between, what is the difference between clistogamy and chasmogamy, so you can check out the links in the description. That is the chapter number 1 playlist link I have mentioned, and you can check out from that link. That is the, what is the difference between clistogamy and what is the difference between clistogamy and chasmogamy with their example. Okay, so this is the question. Next is, explain when a seed of an orange is squeezed, many embryos instead of one is observed. Okay, so when we cut out or squeeze the orange, we have different embryos seen in that structure. Explain how it is possible. B part is, are these embryos genetically similar or different? So if we see the polyembryony, this is because in the orange the polyembryony takes place, more than one embryo is seen. Okay, these kind of structure is seen, that first is the zygotic embryo, second is nucellar embryo, integumentary embryo. Okay, so these all kinds of embryo, more than one embryo is seen in the orange. So the answer of this question is, due to the presence of polyembryony in the orange, the multiple or the many embryos have seen in the structure. Okay, are these embryos genetically similar or different? So if the polyembryony, answer is, if the polyembryony occurs due to the fertilization of more than one egg, then the formed embryo will not be the same genetic constitution. Okay, so if the polyembryony occurs due to fertilization of more than one egg, then the embryos are genetically different. But if all the embryos developed directly from the diploid cell, then they have the same genetic makeup. This is the answer of question. Okay, so the polyembryony, apomixis, all terms are very important. Next question is, what is apomixis? What is apomixis? We have studied in the classes that it is the mode of reproduction which does not involve formation of a zygote through the gamete formation. It is said to be apomixis is a type of asexual reproduction. Yes, it is a form of asexual reproduction that mimics the sexual reproduction in which seeds are produced without fertilization. Okay, so in asexual reproduction, fertilization is not happens. So in apomixis also, the fertilization is not there. That's why it is a type of the asexual reproduction. How is phenomena useful to the farmers? So it helps in the hybrid seed production. Second is one of the most cost effective method to produce seeds, and next is it prevents the loss of specific character in a hybrid. That's why by these all characters it is helpful for the farmers. Okay, so next question is, what is the difference between albuminous and ex-albuminous seeds? So we know that albuminous seeds possess the endosperm. Ex-albuminous seeds, they lack the endosperm. Examples are also very, very important. Okay. Parthenocarpy and parthenogenesis. Example, parthenocarpy is the development of carpy, is the development of fruit without fertilization, and parthenogenesis is the development of new organism without fertilization of an ovum. So the difference is very, very important. Difference between true and false fruit. We have discussed in the previous classes. True fruit develop from the thalamus. Sorry, true fruit develop only from the ovary. Examples are mango, grapes, figs. False fruits develop from thalamus or other floral parts. Example is strawberry and the apple. Okay, apple and strawberry are the false fruit. Why? Because it does not develop from ovary, but they are developed from the thalamus. Okay, so all the differences are important. 19th question is LS of maize grain. This structure of the maize grain is important. Have asked in your 2019-2022. So all the parts is important, and structure and the labeling is also very important. What is the function of the pericarp, endosperm, scutellum, coleoptile, coleorhiza difference is important. This structure is important. Okay, so coleoptile is the covering of plumule and coleorhiza, it is the covering of the radicle. Okay, so this structure is also important. So revise it carefully. Last question of this lecture is that have asked in your 2023, that is the from which end of the ovule, how does the pollen tube grains enter its entry into the embryo sac of the hibiscus plant. Hibiscus is the angiosperm, that is having the flowers, hibiscus flower. So the entry is this structure is important that we have discussed. State the fate of the male nuclei present in the pollen tube. So we know that pollen tube contains 2 male gametes. In this, one male gamete fertilized with egg and form the zygote, which is diploid, and one male gamete fertilized with the polar nuclei that forms the endosperm, which is triploid. So double fertilization process is the answer. Okay, so by this lecture you all have the idea that how the diagrams of this chapter is very, very important. So the diagrams, their labelings, differences, some concept based questions, the outbreeding devices, artificial hybridization, these all topics are repeatedly asked, and diagrams are also very, very important. Okay, so I hope this lecture is beneficial to you all. Okay, if you like this lecture, like, share and subscribe my channel, that is PrepBiology.